Thanks for joining us for today's webinar, Learning Biology at a High Dimensional Scale, presented by Dr. Vivek Das. Dr. Das has been quite the wanderer, having traveled to many parts of the world while earning his bachelor's, master's, and Ph.D. Now, he is currently in the U.S. pursuing a postdoctorate in bioinformatics, studying the importance of biomarkers in target discovery for therapeutics. Please note that the content of this webinar reflects the author's personal views and does not represent his employer in any capacity. With that, I'll hand everything over to Dr. Das to begin the presentation.
Thank you very much for the kind introduction. A warm welcome to everyone. Today I will give a talk about learning biology at a high dimensional scale. Before deep diving into the science, I would like to introduce myself. I was born and brought up in the city of Joy, Kolkata, back in India. As you can see from the map, or well, let's assume for now we all can see it. On the right is my career graph over the years. I've traveled quite a bit around the world to chase my passion. I have a bachelor degree in biotechnology, followed by a master's in bioinformatics from India. After that, I had a short stint of 2 years in the software industry as a programmer analyst. However, my curiosity led me to pursue a Ph.D. in cancer computational biology in Italy, where I also did a brief postdoc. Until I moved to US, where I'm currently pursuing my industrial postdoc. Now let's move forward with the talk. I am primarily dividing my talk in 3 paths, mostly on the lines of Simon Sinek's Power of Why. However, the central subject to my talk is high-dimensional biology. In the first part I will speak about why high-dimensional biology, followed by how do we interrogate it, and finally ending by what do we do with high-dimensional biology. Why high-dimensional biology? Biological systems carry out a lot of tasks that are part of multiple processes. These require massive collaborations in order to give way to a large complex set of processes in our human body and carry out different physiological functions. Thus, it creates a massive wealth of information. This is also very well portrayed in 2007 paper of John Quickenbush, where biological systems have been compared to information management system. They undergo multi-level interactions and influence many events. With high-throughput omics of any kind, such information are getting more and more abundant. These are exposing us to various levels of systems biology, its underlying regulatory mechanisms and circuits. With all such collective information, we are just learning more about human developmental and disease biology, but with more granularity, thus giving us more cues to understand the genotype to phenotype relationship. As a matter of fact, this is also enabling us to access underlying information of various organs, tissues, cells at the scale of systems biology, so that we can tease out the underlying regulatory relationship via a network of DNA, RNA, protein, metabolites, etc., and understand how they function or work via multi-omics avenues. These information, if integrated in a robust model, can lead to better mechanistic insights at molecular level and hopefully bring out potential therapeutics. Now this slide, I provide a few examples of high-dimensional biology. They are used in population genetics and genomics, developmental biology, and disease biology. Now I will walk everyone through the journey of analytical space in high-dimensional biology and how do we interrogate such data. But before doing so, I want to take some time to explain how do we define something in the realm of high-dimensional biology. In my experience, this can be explained via a number of features denoted by P across number of statistical observations denoted by n, the product of which D gives us the informed dimension. Based on these information, as you can see in the image, we usually make underlying assumptions and accordingly employ analytical models like Bayesian, frequentist, or higher order deep learning. This is important as any issues encountered in this step can subsequently impact our data analysis and interpretations, as we can run into the problems of dimensional decurse, overfitting, issue with linear versus nonlinear structure, etc. In the end, this will impact our generalizability and our biological interpretations. Now let us try to visually represent high-dimensional data. In this image, I'm trying to represent different forms of omic profiles comprising of transcriptomics, epigenomics, and proteomics that are often measured together to predict phenotypic contribution. One can see each layer of omics and how they build the overall information space and share relationships. The global or the local representation of them are done here using principal component analysis in the current example. It is a pretty famous dimension reduction method to understand the variability in a reduced space. The top image represents all the omics, while the bottom image represents the correlation across pairs of omics in a multivariate space. Moving on, such representation can also be thought in the single-cell omic space. This image is from a recent Fate paper from Smita Krishnaswamy lab that tries to represent various cell types and its underlying latent structures. Moving from left to right, we can see various cell types that are profiled. These are measured and quantified via high-throughput platforms, and their overall representation is in the form of a high-dimensional matrix as cell cross-gene, which is the middle image. Finally, we then visualize this using state-of-art method that guides us and helps us to learn various biological states, lineages, and transitions for any interpretations and inferencing, as you can see the third image. Hope I could provide a flavor of high-dimensional biological data. Now let us learn things needed to interrogate such data. First and the foremost is the requirement of a platform that can perform such interdisciplinary science adhering to FAIR principles. Once we have a platform in place, our next task is to build a robust integration system or a workflow. This should comprise of various state-of-art statistical and machine learning tools so that we can ingest, analyze various forms of omics data of any dimensions, either in a singular manner or integrated, to bring out predictions, pathways, networks, biomarkers that can reach us more closer to our goal of phenotypic understanding. Now let us try to understand what the strategies employed or used in bulk multiomics. Let's run the processes used in interrogating high-dimensional biology. In my experience, this is not a simple task. It needs knowledge of domain expertise over here biology of interest, statistics, mathematics, computer science, etc. Make use of all of them together to study the overall data property, structure, behavior, as you can see here, thus aiding us to gain enough knowledge to predict phenotypes and bring out potential future therapies. However, in performing such, we have a lot of challenges in our path. As you can see from this slide, there are various challenges that are portrayed by Misra et al. In their 2019 Integration Omics review. Each of these needs to be tackled in order to make robust biological interpretations from massive high-dimensional data. From an analytical perspective, two problems that usually need utmost attention are individual omics, datasets, and integration issues as highlighted. One should take a lot of attention and care during these steps. One of the simplest way to tackle is by investigating the features or the underlying variables that are used to make any phenotypic prediction. If we can identify, extract, and select them well using robust analytical methods, our prediction will be less affected. To this end, we will need to take the pursuit of statistics or machine learning given the overall nature of the data and problem scope in hand. Having said that, I feel we are already in an advantageous stage as a lot of seminal works have been carried out thanks to our experts who have already built a suite of multiomics methods that can take care of such data and multiomics integration issues. These can be categorized in two ways, as you can see in this slide. One that is statistical and the other that is machine learning or deep learning based. Now, as you can see, most of these methods perform specific functions as outlined in different boxes. Such are reliant on the data properties, structure, and queries one needs to address while using high-dimensional multiomics. Given the data order is huge and massively heterogeneous, one can also take assistance of machine learning and deep learning. However, something I want to stress here, as per my experience, machine learning and deep learning is powerful, but it is beneficial mostly when the underlying data and the problem scope has enough bandwidth to fit such models. Another important to note here is some methods under the specific categories do overlap under statistics and machine learning. Now in this slide, I'll give an example of such multiomics integration at high dimension that we published a few weeks back with researchers from India. Our goal was to find out if gene expression is influenced by DNA methylation and copy number changes at high dimension. We used the Cancer Genome Atlas liver hepatocellular carcinoma datasets as a model system in order to establish gene expression changes as a function of DNA methylation and copy number changes. We pursued feature extraction using deep learning strategy like denoising autoencoder. This was followed by regression using multilayer perceptron to estimate the overall gene expression pattern. Now here is the final model, as you can see here. Then we benchmarked this state of art with other regression methods to understand how well and robust our current predictive model fares in comparison to the others. And we did this at various regularizations. One of the key motivations to use deep learning was to see whether our method can capture the underlying nonlinear relationship and events as often seen via the stochastic behavior of gene expression. Now let us understand what are the strategies used in single-cell omics. So far, I have demonstrated methods to integrate mostly multi-omics, high-dimensional biological data with respect to bulk profiling of samples and tissues. However, we know currently that we can also add a new layer of information, that is individual level cellularity via single-cell technology. I will not go into much details about the technological innovation, but I will try to address how are we placed on the analytical side of it. Now, this screenshot, as you can see, is from the website scrnsectools.org, and it represents the current number of tools that are currently available in different categories. As you can see, it's over 600 tools already if I look at the left map, and it's over 30 categories. This database was created by Oshlak Lab based in Melbourne, Australia, and currently maintained by a team of volunteers around the world at Institute of Computational Biology in Munich. Now let us try to understand what are the processes of integrating the single-cell data. Well, we are in fact using diverse methods in the single-cell analytical space. In the last few years, many amazing innovations and discoveries have been made on that front. Thanks to the effort of many, that our current high-dimensional analytical methods are being adapted to analyze even single-cell data. We have come a long way with new tools being developed on this front. Such works have paved the way to some of the best practices in the single-cell analytical space, as you can see in the form of this image. These can aid at various levels of data analysis pertaining to normalization, batch correction, clustering, dimension reduction, etc., that we often encounter at various stages of single-cell data analysis. These are all nuts and bolts that are required to bring out the effective single-cell analysis for deriving robust results, which can aid for biological interpretations. However, it is important to ask, did we solve all the problems? As we know, with single-cell data, the dimension is often pretty high, and the number of data points are pretty higher than the traditional sequencing data. My answer to this query is no. We have come a long way with more data generation, but we are also getting aware of more and more challenges in this space. One such example will be the recent community effort that led to the publication of 11 grand challenges in the single-cell data science space. This informed us of areas that need attention, some of which is outlined below, like that of the data sparsity, flexibility in statistical frameworks that needs to be understood for capturing differential expression, data missingness issues, data integration across samples, experiments, and various types of omics measurement. Now, how can we address these challenges? In my view and experience, this serves as an amazing opportunity. These challenges can be actually tackled given we pursue systematic benchmarking efforts. As we are generating a lot of data and tools in this space, we also deposit them publicly in several platforms. Thus, if we employ systematic benchmarking efforts, we can narrow down robust methods in this space. However, I would like to mention that there's a lot of progress lately in the benchmarking front in single-cell field. This field is very active. Such benchmarks are not only restricted to the single-cell technology and platforms, as you can see from these two papers, they have also been extended to the analytical space such as dimension reduction and visualization of such, even to cell type identification and many more. This space will be very interesting in the upcoming years to see how the field matures and progresses while tackling complex analytical challenges using varied forms of high-dimensional data. Finally, I'm coming to the end of my talk. What do we do with high-dimensional biology? Well, in this slide, I will cite a few examples. Some of it are large consortia like the Cancer Genome Atlas or ICGC that educated us about molecular bases of various cancer at a scale. Then databases like Omics Discovery Index that works as an aggregator of various other multi-omics consortium and databases, only to help us map tissue-specific molecular information across diseases in various organisms, thus providing us more mechanistic insights of the underlying biology. Finally, I want to cite the Human Cell Atlas efforts. This is taking to a next level where multi-omics data is being mapped not only at organs and tissue level in humans, but we also get the information at the individual cellular level. a tremendous community-driven effort promising to leverage biological knowledge and data together, thus enlighten us with developmental and disease biology at a cellular level granularity. Now in this slide, I would like to present there are already some pretty exciting databases and portals available for the community for further exploration. So this is a database, the Shiny app that was developed by Pranit Chaturvedi, which actually lists all the available free portals. And as you can see from the sources, I really want to appreciate the efforts of the research community for sharing this with all of us. Here are some of the publicly available resources, like that from the Broad Institute, from European Bioinformatics Institute, from UCSC, that can be utilized for quick exploration. What we can do is we can explore a bunch of genes, identify its underlying putative cell type associations in organs and tissues of interest, and it can help us with the hypothesis establishment. This is another area of application of high-dimensional biology. Finally, in my last slide, I'm trying to put an overall application and perspective of high-dimensional biology in the era of omics. As per my understanding, it is trying to unify and integrate information from various arms of biology like molecular, spatial, anatomical, etc. Thus, provides us with a multi-organ, multi-tissue toolbox that is also extended to the single cell, given we can integrate the data well with robust methods, thus making us better at understanding cellular phenotypes. In doing so, the aim is to get better at understanding various domains of systems biology, get improved understanding of pathophysiology, and discover potential new therapies as represented in this figure. I have come to the end of my webinar. Before signing off, I want to acknowledge each and every one who have over the years supported and guided me in every possible manner. I thank the entire research community who provided me with all the learnings to critically think and eventually get a little bit more clarity in the high-dimensional biology. Hope I could impart some of this knowledge today via this webinar, and I'm happy to take questions.
Great. Thank you for that insightful presentation, Dr. Das. We did have quite a few questions trickle in, so let's jump into those. The first question we received is, where do you see the field moving?
Thank you for the question. That's actually a great question. I mean, for now, these are very early stages, and I totally see that it can really help us in understanding the biology at a scale and eventually give us an understanding how we can better understand the human pathophysiology, given we actually able to get better at analyzing the data. But there is a lot of potential I see in this, specifically with respect to therapeutics, finding new biomarkers, new targets that can be then tested in clinics. This is how I see, but we need to do a lot of groundwork for there to reach. This is my answer for this.
Great, thank you. The next that came in was, how important is biological domain knowledge while performing such high-dimensional biological data interrogation?
That's another fantastic question. Actually, I always think that biological domain knowledge is very important because when we use data, we also need to know what are the critical questions we need to ask with this data, right? Otherwise, we will get a bunch of variables and we will predict something, but the context can be missing. And biology is very noisy. So we need to actually know our specific contextual questions. And this is where domain knowledge comes into handy, because our goal is to make us understand where the domain knowledge of biology and the data-driven knowledge can converge for us to either make new hypotheses or actually align with the hypothesis which we started with. This is where I think both biology and data comes in handy, and domain knowledge serves as a very important piece of information.
Another we received was, is there a generalized method that can tackle most of the analytical challenges?
Well, as per my experience, this is very tough. It's not very straightforward. There cannot be a generalized methods because all these things are reliant on the kind of data we interrogate. And this is also very problem specific. Simple models are there, but that does not mean that those simple models will handle everything. It is not very easy to say because whenever we interrogate very noisy or heterogeneous data, especially in the realm of high-dimensional biology, we end up getting to clean up the data a lot, trying to understand the relationship in the data. And that is often not possible with simple models or with the default models. We need to do a lot of tweaking of the parameters. And for me, That's where these kind of regularization we use or parameterization we use that can say that there is not possibility of having a generalized method. But that does not mean that their simple models or general models won't work. They will work, but there will be different levels of uncertainty to whatever we find out.
Next is, what method is superior for multiomics integration analysis? Is it stats-based or ML-based?
This is another fantastic question. In my experience, the kind of data sets I have been integrating or have been integrated, it's not a straightforward approach. I'm a person who enjoys where both converge together. And there are a lot of methods that I have been using lately where the statistics and the machine learning converges. There is no one-size-fits-all theory, as per my understanding. So given the data nature, the data property, and the behavior, and the query we are addressing, it should be able to guide us and help us which should be the method that we should be using, whether it's the statistical based or whether it's a machine learning based. And as I said in my presentation, the usage of machine learning and deep learning is only beneficial when our data points out the complexity in our analytical space. So both are equally important. Which will be the top one, it's very difficult to say given what we are trying to address, because this will be very much problem specific. There are methods where we actually start with statistics, and given that the problem complexity goes higher, that is when we take aid of machine learning and deep learning. So I would say we will be using both, and it will be very difficult to address whether one will be outperforming the other.
Another is, what are the current available resources for analyzing or interpreting high-dimensional biological datasets? Are there free websites to perform any exploratory analysis?
So this is a great question again. So I have listed out in one of the slides what are the different methods that are currently available. If there are two fantastic publications that have already shown what are the different categories and what are the different methods that have been employed to actually integrate multiomics data, be it using statistical or be it using machine learning or deep learning. And these can be integrated when we use any kind of analytical softwares like open source R Studio or R server. One can install them freely in the computer and do the analysis. There are also Jupyter notebooks which are Python based or R based where one can perform all this analysis. And Bioconductor and CRAN are the repositories where we can download a suite of different kinds of methods if we are using R. If we are using Python, there are different packages that are available in sklearn toolkit, which can help us doing all these kind of analysis. These are the resources point. As for the free websites, well, I have listed some of them like TCGA and ICGC data portals. For the single cell, I would really point out the resources that are available from UCSC, European Bioinformatics Institute, and Broad Institute. These are all datasets that have been currently been published, and they are being deposited in these websites where anybody can actually go and look up for a bunch of genes and try to understand what is the associated putative cell type. The funny thing is, I actually did a short survey in Twitter a few days back and tried to understand how many people are aware of these resources, and it came out that only 62 people actually voted there in my poll, and out of which over 40% did not know that some of these resources are freely available. So I hope with this presentation I'm able to actually provide some sort of learning that there are already certain amazing resources available for everyone to explore. It is not always that we have to analyze using complex methods. There are some state-of-art availability of tools that can also help us derive, explore, and provide new hypotheses.
Just one last question here. What is the computing infrastructure needed to perform these analyses?
This is a very nice question. Well, I mean, we can say that we need a very high performance computing grid or servers. But there are some very nice avenues that I've seen that are currently working beneficial at my end. I will just say what I have been using. So I have a Mac here at my end, and it's not very highly computationally strong. But for certain analysis on the projects which I do, I am able to use my Mac here. But to make a computationally intensive model, it is important that we have a large amount of processing unit. For this, I'm using the Google Colab. I'm finding it very beneficial at my end because we are actually entering into a cloud platform that has all the required resources that we need for running any kind of computational intensive analysis. As far as my understanding is concerned, it is entirely free and one can actually set up via their own logins, Gmail IDs, and they can launch any kind of cloud sessions from there and perform any kind of exploratory data analysis. I won't be saying that we can do method development or continuous integration development. I have no experience on that, but creating exploratory data analysis and notebooks in Python and R for any kind of model exploration, I have found them pretty convincing enough at my end to run any kind of models, and I have been really having a lot of fun using them, and they are completely free resources.
Once again, I'd like to thank Dr. Vivek Das for his presentation and all the attendees for joining us. Thanks for watching.